Most of the world didn't have it until colonization started in the 15th century.
And it was discovered that the rhesus factor could be positive or negative. Most humans in the world are rhesus positive.
For over a century, doctors have quietly relied on a piece of biology so useful it borders on miraculous: type O blood, the so-called universal donor. It sits in blood banks around the world, ready to be given to nearly anyone in an emergency, regardless of their own blood type. Most people who have donated blood or received a transfusion have encountered this fact without ever asking why it's true or where it came from. But when scientists began digging into the deeper story of type O blood, tracing it across continents, back through ancient DNA, and into the mechanics of disease itself, they found something far stranger and more consequential than a simple medical convenience. They found a signature of survival, written into human genetics by thousands of years of exposure to some of the deadliest diseases our species has ever faced. The story begins, as so much of modern genetics does, with a discovery made before anyone understood DNA at all. In 1900, an Austrian physician named Karl Landsteiner was trying to solve a mystery that had puzzled doctors for decades. Why did blood transfusions sometimes save a patient's life and other times kill them almost instantly? Landsteiner began mixing blood samples from his colleagues and himself in test tubes, and he noticed that some combinations clumped together violently while others mixed smoothly. From this simple observation, he identified the existence of distinct blood groups, what we now call B and O. A colleague of his later added the fourth group, AB. For this work, Landsteiner would eventually win the Nobel Prize, and his discovery would go on to save more lives than almost any single finding in the history of medicine. It became possible for the first time to know in advance whether a transfusion would heal a patient or destroy them. What Landsteiner had actually found was a difference in the surface of red blood cells. People with type A blood carry a specific sugar molecule, called an antigen, on the surface of their cells. People with type B carry a different one. People with type AB carry both, and people with type O carry neither. This absence is the entire secret behind type O's universal donor status. The immune system attacks foreign antigens it doesn't recognize, which is why giving type A blood to a type B patient can trigger a catastrophic immune reaction. But type O blood has no A or B antigens to trigger a response. It slips past the immune system's defenses because, in a sense, it has nothing to be recognized as foreign. This is why type O negative blood, in particular, is kept in reserve in every major trauma center and battlefield hospital on Earth. When there's no time to test a patient's blood type, type O is the safe bet. For decades, this was treated as a fairly simple biological quirk, useful for medicine, but not much else. But as genetic research expanded beyond hospitals and into the study of human population history, researchers began to notice something curious. The distribution of blood types across the globe is not random. It's not evenly spread. In parts of Europe and Asia, all four blood types exist in substantial numbers. Type A is especially common in parts of Scandinavia and Japan. Type B shows up more frequently across Central Asia. But when researchers turned their attention to the indigenous peoples of the Americas, they found something remarkable. Type O blood didn't just dominate. In many populations, it was almost the only blood type present at all. Studies of blood type frequency among indigenous groups in Central and South America have repeatedly found type O frequencies above 90%, and in some isolated Amazonian and Andean communities, close to 100%. This is an extraordinary level of genetic uniformity for any trait, let alone one tied to something as fundamental as blood chemistry. For comparison, even in populations known for genetic isolation elsewhere in the world, you would typically expect to find some residual variation, some scattered presence of other blood types surviving in the background. In much of the Americas, A and B alleles are not just rare, they are functionally absent. The first explanation scientists reached for was one of the oldest and most reliable tools in population genetics, the founder effect combined with genetic drift. The idea is straightforward. When a small group of people migrates into a new territory and becomes isolated from the larger population they came from, the genetic makeup of that small founding group, whatever it happens to be, becomes the genetic starting point for everyone who follows. If purely by chance, the group that first crossed into the Americas thousands of years ago carried a disproportionate share of type O alleles, then their descendants would inherit that same skew, and over many generations, with little to no genetic mixing from outside groups, that skew could become even more pronounced. Small populations are especially vulnerable to this kind of random drift because with fewer individuals, chance events, who has children, who doesn't, who survives a hard winter, who doesn't, can have an outsized effect on which genetic traits get passed forward. This explanation is not wrong. Founder effects are real, well documented, and they undoubtedly played some role in shaping blood type distribution in the Americas. The migration of humans into North and South America appears to have occurred through a relatively small population, likely numbering in the low thousands, crossing from Siberia into Alaska sometime between roughly 15,000 and 20,000 years ago, either along a coastal route or through the interior of what was once the Bering Land Bridge. A small founding population, followed by thousands of years of relative isolation from Eurasian and African populations, is exactly the kind of scenario in which genetic drift can produce dramatic, uneven outcomes. But founder effects and drift alone have struggled to fully explain the sheer scale and consistency of type O dominance across such an enormous and ecologically diverse range of territory. The Americas are not one small, isolated valley. They span from the Arctic tundra of Alaska to the tropical rainforests of the Amazon to the high, thin air of the Andes to the temperate plains of North America. Populations across this entire range, separated by thousands of miles and thousands of years of independent history, ended up converging on the same overwhelming type O dominance. That kind of consistency, appearing again and again across genetically and geographically distinct groups, is the sort of pattern that makes researchers suspect something more than random chance is at work. When a trait shows up repeatedly, independently, across very different environments, biologists start looking for a common selective pressure, something in the environment or in disease exposure that actively favored one outcome over the others, rather than leaving it purely to luck. This is where the story connects to one of the most well-established, if still incompletely understood, areas of modern medical genetics, the relationship between ABO blood type and infectious disease. Over the past several decades, a substantial body of research has accumulated showing that blood type is not immunologically neutral. It actively influences how susceptible a person is to certain pathogens and how severely they experience an infection once they have it. The clearest and most extensively studied example involves cholera, a disease caused by the bacterium Vibrio cholerae, which has been responsible for devastating pandemics throughout human history and remains a serious public health threat in parts of the world today. Cholera kills through severe dehydration, caused by a toxin that the bacterium produces once it colonizes the small intestine. Research has shown that this toxin interacts differently with the different ABO blood group antigens found in the cells lining the gut. People with type A or type B blood tend to experience significantly more severe cholera symptoms than people with type O blood. In some studies, individuals with type O blood who contracted cholera had a substantially lower risk of developing the most severe, life-threatening form of the disease compared to those with type A blood. In a world without modern rehydration therapy and antibiotics, a difference like that would not be a minor statistical curiosity. It would be the difference between surviving an outbreak and not. A parallel story emerges with malaria, one of the most consistently lethal diseases in human history and a disease that has shaped human genetics more dramatically than perhaps any other, evidenced by the persistence of traits like sickle cell trait despite its costs, because it offers protection against the parasite. Malaria is caused by parasites of the genus Plasmodium, and the most dangerous form, caused by Plasmodium falciparum, kills through a process where infected red blood cells become sticky and clumped together inside small blood vessels, a phenomenon called rosetting, which can block blood flow to vital organs including the brain. Research has found that this rosetting process happens far less efficiently in people with type O blood than in people with type A, B, or AB blood. The absence of the A and B antigens on the surface of red blood cells appears to make it physically harder for infected cells to stick together into these dangerous clumps. Multiple studies conducted in malaria-endemic regions of Africa have found that people with type O blood are significantly less likely to develop severe, life-threatening malaria compared to people with other blood types, even though they can still be infected and can still get sick. When you put these two disease relationships together, cholera and malaria, a picture starts to emerge that goes a long way toward explaining why type O blood might have spread and persisted so dramatically in certain populations without needing to invoke anything mysterious or unexplained. Populations that endured repeated waves of severe infectious disease, whether cholera-like illnesses, malaria, or other pathogens that interact with blood group antigens in similar ways, would have experienced real, measurable survival advantages for individuals carrying type O blood. Over many generations, in populations facing intense and repeated disease pressure, even a modest survival edge can compound dramatically. This is simply how natural selection works. It doesn't need a single catastrophic event to reshape a population's genetics. A repeated, small advantage, generation after generation, can eventually produce populations where the advantageous trait becomes overwhelmingly dominant, especially when combined with the amplifying effects of a small founding population and genetic isolation. This combination, founder effect plus sustained selective pressure from infectious disease, offers a coherent, evidence-based explanation for what researchers observe in indigenous populations of the Americas without requiring speculation about hidden conspiracies, buried research, or secret genetic switches. It's a story that fits comfortably within everything else we understand about how evolution shapes human populations in response to their environment. It also helps explain why type O prevalence is not uniform even across indigenous American populations. Groups with different disease histories, different population sizes at founding, and different degrees of later genetic mixing with European, African, or Asian populations following colonization show correspondingly different blood type distributions today. The story of ABO blood type and disease resistance doesn't end with cholera and malaria either. Researchers have also investigated connections between blood type and a range of other conditions, from certain types of cancer to cardiovascular disease to, more recently, COVID-19. Several early studies conducted during the COVID-19 pandemic found statistical associations between blood type and both the likelihood of infection and the severity of illness, with some research suggesting people with type O blood may have had somewhat lower rates of severe outcomes compared to people with type A blood, though this research area remains genuinely contested with some later, larger studies finding much weaker or inconsistent effects once other factors were properly accounted for. Scientists are still working to understand exactly which biological mechanisms are responsible and how strong or consistent these effects really are across different populations and variants. This is worth sitting with for a moment because it illustrates something important about how real science works. The picture is rarely as clean or as dramatic as it first appears, and researchers are often left revising their conclusions as more data comes in, rather than arriving at a single, tidy, sensational answer. There's also a deeper evolutionary story behind blood types that stretches back long before humans existed at all. The genes responsible for the ABO blood group system are extremely ancient, far older than our species. Related versions of these genes have been found in other primates, including chimpanzees and gorillas, suggesting that this genetic system has been shaped by selective pressures, likely including infectious disease, for millions of years across multiple branches of the primate family tree. This tells us that the relationship between blood type and disease resistance isn't some strange, recently discovered anomaly. It's a genuinely ancient evolutionary arms race between our immune systems and the pathogens that have hunted humans and our ancestors since long before recorded history, one that has left visible fingerprints in our DNA that scientists are still working to fully decode. None of this means type O blood is some kind of superior or chosen genetic type, an idea that would badly misrepresent how evolution actually works. Every blood type persists because in some context, in some environment, facing some particular set of pathogens, it offered enough of a survival advantage, or simply wasn't disadvantageous enough, to be passed down through the generations. Type A and Type B blood haven't vanished from most of the world's population because they, too, carry their own resistances to different diseases in different environments. There's research suggesting, for instance, that Type O blood may actually carry a slightly elevated risk of certain conditions, including some studies linking it to higher rates of peptic ulcers associated with H. pylori infection. Evolution doesn't produce a single winning trait. It produces a shifting, context-dependent balance of trade-offs, where what helps you survive one disease might leave you more vulnerable to another. The overwhelming dominance of Type O in the Americas likely reflects a set of disease pressures and a founding population that happened to align in a way that made this particular trade-off favor Type O blood especially strongly in that specific historical and ecological context. What makes this story genuinely fascinating, rather than something that needs artificial embellishment, is what it reveals about the relationship between our bodies and our history. Every one of us is walking around with a genome that has been shaped, quite literally, by which of our ancestors survived epidemics that we will never know the names of, diseases that may not even exist anymore in the form that once threatened our species. Blood type is one of the clearest, most measurable windows we have into that deep history. When researchers look at a blood sample and see an overwhelming skew toward Type O in a particular population, they are looking at the accumulated outcome of thousands of years of survival, illness, and reproduction. They are looking, in a very real sense, at a message written by the people who lived through diseases we can no longer even identify, encoded not in language or in written record, but in the literal chemistry of their blood and passed down to everyone who descended from them. This is also why modern genetic research into ancient DNA has become such a valuable tool for understanding this history more precisely. As techniques for extracting and sequencing DNA from ancient skeletal remains have improved dramatically over the past 2 decades, researchers have been able to directly examine the blood type alleles carried by people who lived thousands of years ago, rather than relying solely on inference from present-day populations. This kind of ancient DNA research has confirmed that populations in Siberia, the region from which the ancestors of indigenous Americans are believed to have migrated, showed a genuinely diverse mix of blood types in the deep past, much like most of Eurasia does today. This supports the idea that the overwhelming typo-dominance seen in the Americas developed after the migration, shaped by the combined forces of founder effects and disease pressure acting on the descendants of that founding population over thousands of subsequent years, rather than existing before the migration ever took place. It's worth pausing to appreciate how remarkable it is that any of this can be reconstructed at all. A blood sample drawn from a hospital patient today, a routine part of a checkup or a donation, carries within it traces of events that occurred long before written history began, outbreaks that swept through ancient communities, generations of people who lived and died based partly on a molecular difference on the surface of their red blood cells, and a slow, patient process of natural selection working across thousands of years. Typo blood earned its place as the universal donor not through some hidden agenda or forgotten conspiracy, but through a very real, very human story of survival against disease, written by evolution itself and preserved for us to read, quite literally, in our own blood. The next time you see a blood drive or hear that hospitals are always in urgent need of typo donors, it's worth remembering that this small vial of donated blood carries an extraordinary history within it, one that connects a modern donor to ancestors who endured epidemics we can barely imagine, and whose survival, quite literally, made possible everyone who came after them. Science hasn't finished uncovering everything this story has to teach us. Researchers are still working to understand exactly how strongly blood type interacts with newer diseases, why certain associations appear in some studies and not others, and what other secrets might be hidden in the genetic variation we've inherited from our ancestors. But what we already know is compelling enough on its own. Our blood is not just a medical classification. It's a record, imperfect and still being decoded, of everything our ancestors survived to bring us here.